So let's take a look at this action potential and the means by which we can propagate or conduct the signal through the system. And so we have a distinct sequence of events within the opening and closing of voltage-gated channels based on the relative and absolute refractory periods, the depolarization and hyperpolarization moments, where we have to reach threshold at the axon hillock. This is the initial segment that starts the propagation. This is the terminus of the localized sum graded potential. After the axon hillock, it is strictly action potential if we reach threshold.
The fundamental task of a nerve cell is to receive, conduct, and transmit signals. Neurons propagate signals in the form of action potentials, which can travel great distances along an axon without weakening. To transmit an action potential over such a distance without weakening requires that the signal is continuously reamplified along the way. The central molecular players in this process are the voltage-gated sodium channels, which undergo a cycle of finely choreographed conformational changes. When an action potential passes, sodium channels open in response to membrane depolarization. Sodium ions rush into the axon, further depolarizing its membrane. Within a fraction of a thousandth of a second, however, the sodium channels switch to a new inactivated state, in which they are closed, but now also refractory to reopening. In this way, the membrane potential can recover quickly after an action potential has passed. The sodium channels then reconvert to the closed state, ready to be opened again when the next action potential is encountered. Let's examine the changing state of the sodium channel during an action potential. When no stimulus is present, the sodium channels remain closed, and the electrical potential measured across the membrane remains constant. However, if a depolarizing stimulus is applied by a brief pulse of electric current, the membrane will start to depolarize, away from the resting potential of about -80 millivolts. Some of the sodium channels will open, permitting sodium ions to enter the axon along their concentration gradient. If the depolarization is sufficient, even more sodium channels open, and the membrane potential rapidly approaches the equilibrium potential for sodium, about +40 millivolts. At this point, the sodium channels close, adopting the inactive conformation, where the channel is unable to open again even though the membrane potential is still depolarized. The sodium channels will remain in this inactivated state until a few milliseconds after the membrane potential returns to its initial negative value.
So let's take a look at how this functionally looks for the voltage-gated channels. So we start with the initial stimulus. The initial stimulus will open up the sodium channels, allowing for sodium to influx rapidly into the cell, taking it into a depolarized moment. This leads to the opening of the potassium channels and the inactivation of the sodium channels. This leads to the efflux of potassium and no movement of sodium whatsoever. This slowly leads to the reestablishment of the membrane, in the reconfiguration of the sodium channel back to its resting state. Notice the potassium channel is still open, and this is where the sodium-potassium pump will come into play, attempting to reestablish the resting membrane potential, bringing it from a hyperpolarized state back to the resting state where both voltage-gated channels are closed. We do not get a larger ion flux just because the stimulus is larger. What we can do is we can get through the refractory period slightly faster, leading to a greater number of action potentials, but not a larger action potential. So the impulse conduction is going to start at the axon hillock if the axon hillock reaches threshold. It will then be conducted through what's referred to as local current flow through the fiber. This local current flow is impacted by two things. One, the size of the axon, based off of what's referred to as Henneman's size principle. And two, the absence or presence of myelin. Based off of the absence and presence of myelin, we get two types of impulse conduction. We get what's referred to as continuous conduction on an unmyelinated fiber. That is where each segment, each micrometer of the axon has to undergo depolarization, repolarization, hyperpolarization, resting before it can go about being depolarized again. The other means of doing this is what's referred to as saltatory conduction. Saltatory conduction only occurs on the myelinated fibers, in which areas of the in-between Schwann cells at the nodes of Ranvier are the only areas that have to undergo the full cycle of action potential. So in continuous, we go segment by segment by segment, all the way through. Notice how far the signal has gotten in 10 milliseconds.
An action potential, depicted as a red band, is propagated in one direction along the axon. During an action potential, the inside of the cell membrane becomes positive with respect to the outside. An action potential generates local currents that tend to depolarize the membrane immediately adjacent to the action potential. When depolarization caused by the local currents reaches threshold, a new action potential is produced adjacent to the original one. Action potential propagation occurs in one direction because the recently depolarized area of the membrane is in absolute refractory period and cannot generate an action potential.
What's it going to look like? And so because the aft region of the membrane closest to the soma, is in refractory, the only area that can undergo the depolarization moment to threshold.
The improved efficiency is due to the fact that the insulation provided by certain cells, called Schwann cells, which have wrapped themselves around each axon effectively limits the movement of ions to short segments between these cells, rather than allowing this ion exchange to take place along the entire length of the nerve fiber. Loss of the myelin sheath can cause serious impairment of function of the nerves involved.
Is towards the terminal ends. Within the propagation, the second means by doing this is with saltatory conduction. In saltatory conduction, we don't need the entirety of the membrane to undergo depolarization. While myelin can allow for the speed of propagation to be faster, the propagation speed is not related to the strength of the stimulus. The neurons will propagate the signal at the same rate. However, different neurons will propagate at different speeds. This has to deal with the amount of stimulus that is there and the amount of membrane that has to take place. So what ends up happening is that with large axons, I can propagate much faster than small axons. I can propagate much faster with large axons relative to small axons for two reasons. Number one, I need more stimulus in order to get to threshold, which means I have more energy being dissipated. Number two, I have more membrane that is undergoing a response, which means I have greater voltage change, allowing for greater depolarization to occur. However, I need more stimulus there in order to get that larger membrane to get to threshold. That is Henneman's size principle. The speed and threshold are proportional to the size of the axon. It takes more stimulus to get a plus 15 millivolt change in a large neuron than it does in a small neuron. The other thing that happens with large neurons is because of the amount of axon that's there, they are more likely to be myelinated than the small axons. Because large fibers are myelinated, they undergo saltatory conduction. And since saltatory conduction is faster than continuous conduction, they will propagate the impulse at a faster speed. In this, we have different fiber types with different thresholds and different speeds. The largest is the A fibers. They have the largest threshold. They are the largest fibers at 5 to 20 microns in diameter and can propagate up to about 130 meters per second. We have medium-sized fibers, which are somewhere between 2 and 3 microns and will propagate at around 15 meters per second. And then we have the smallest fibers, which are between a half and a micron and a half in diameter and will propagate at about 2 meters per second. You can look at the diameter size and determine the length that the axon will be coming away from those fibers. Where typically small fibers tend to be short in full length, whereas large fibers tend to be much longer in full length. We can pick up the speed of propagation based off of peripheral nerve pulses, where we can actually see and register nerve impulses based off of the propagation, and look at how long does it take for a signal to go from point A to point B on the neuron. There's other ways in which we can measure neuronal activity, and that is centrally. There are two ways in which we can measure neuronal activity centrally. The first way is with an EEG. This is going to measure the electrical output that takes place. The other means by doing it is with a CT scan or a functional MRI. The CT scan and functional MRI are measuring blood flow and fuel utilization, whereas the EEG is measuring electrical output. In the CT scan and in the functional MRI scan, we are measuring areas of hot versus areas of cold. Areas of hot is an indication of large amounts of blood flow. Where we have large amounts of blood flow is an indication of large amounts of activity taking place. Areas that are cold are an indication of areas of low activity, and lower amounts of blood flow heading into those areas. The other means of measuring is through an EEG readout. An EEG readout is going to measure the electrical wave activity taking place via a three-dimensional electrode arrangement in which we have four distinct wave patterns that come about. The alpha wave pattern tend to be slower in frequency, higher in amplitude, with a frequency range of between 9 and 14 cycles per second. If you have completed a task and are sitting there not doing much, you would tend to be in alpha state. Beta state, the frequency picks up and the amplitude shrinks slightly. We go from 9 to 14 cycles per second to 15 to 40 cycles per second. This is an indication that you are actively engaged in a mental activity. The other two wave patterns are the theta and the delta waves. Theta waves, we typically have larger amplitude than the alpha waves and slightly slower frequency than the alpha waves. We're somewhere between 5 and 8 cycles per second. This is where we are not actively engaged with what's going on around us. We are quote unquote daydreaming or in la la land. The delta wave patterns have the greatest amplitude and the shortest frequencies. These tend to cycle between 1.5 and 4 cycles per second. This is an indication that you are in dreamless sleep. Typically during sleep, you're somewhere around 2 to 3 cycles per second. We never see any of the delta wave patterning go to 0. If we see delta wave patterning go to 0, that's an indication of no neural activity, where you are isoelectric across the cerebral cortex, or what is sometimes referred to as being quote unquote brain dead. You just need the areas in between Schwann cells to undergo depolarization. The current flow goes around the Schwann cells. The Schwann cells act as an insulator to ion movement. And because it acts as an insulator to ion movement, the only area where ions have to move is in between the Schwann cells. Notice how much closer we are to the terminus at 10 milliseconds via a saltatory conduction versus a continuous conduction. So notice that when we had the myelin there, we didn't need the entirety of the axon to undergo depolarization, just the small segments. And what this does is it speeds up the propagation because I don't need the entire length of the axon to undergo depolarization.